So this presentation is looking at the Hardy Weinberg principles and the equations and use of equations in the A-level examinations. So to start us off, again, just a couple of terminologies that you should be familiar with. Most of these appear on GCSE. Some of them you may not be so familiar with, but you definitely need to know them off by heart because again, you're going to find it very difficult to pick up marks in exam questions if you don't use these terms effectively. So pause the video at this point and have a go and see what you can come up with for definitions of each of the words and then check through your answers. So a species is a group of organisms that can breed and produce fertile young. A population is groups of organisms of the same species is the important part, but you should include same habitat at the same time. Allele frequency is the number of times an allele occurs within a gene pool. The gene pool refers to the total number of alleles in a population at any particular time. A community is where you have groups of organisms of different species in the same habitat at the same time. And of course, the habitat is where a particular community lives. There is an extension question, which looks at, is the dominant allele more common in a population than the recessive allele? It very much depends on what the phenotype is resulting from the dominant and recessive alleles. Some of the different genetic conditions and stuff that you can find actually are a result of dominant alleles rather than recessive ones. So don't always make the assumption that dominant is more common in a population than recessive. So is it possible to work out the genotypes of everyone in a population just by looking for a particular feature? Well, the complicated answer is no, because you have a changing population with deaths and births, and you'd never be able to collect the data. So from a practical point of view, no. But even more so, if you could freeze frame an entire population in a picture, you still wouldn't be able to work out the genotypes, because if you have a dominant and recessive condition, the dominant heterozygous organisms would look exactly the same as the dominant homozygous organisms. So you wouldn't be able to work out which was which genotype. The recessive homozygous would be easy to spot, obviously, because they look different to the other organisms, but it would certainly be a problem in terms of separating out the homozygous dominant and the heterozygous. So the first thing to note about the Hardy-Weinberg equations is that they are only applicable under a certain set of circumstances. We have to make certain assumptions for these equations to apply. The assumptions are that the population is large, there's no mutation, migration, natural selection, or sexual selection in any type or form. If we take all those concepts away, it's obviously not true to life, but it will let us do some of the calculations and data and give us some of the values that will let us see patterns and trends in different allele frequencies and in genotype frequencies. And again, it ties back to the previous question over, can we get the actual genotypes of every single organism? So the two equations you need are P plus Q equals 1, and that's linked to the allele frequency. The second equation is linked to genotypes, where you've got P squared, 2PQ, and Q squared equals 1. So to start with, we're not so worried with the application of the equation at this point. All we want to look at is the structure and format that's been set out in this particular answer. The information we're given is the phenotypes of the organism, and you can see clearly that they've used the letter Y. Not the best letter, but they have distinguished dominant and recessive quite clearly, so it will work. You've also got the genotype frequencies. Note that genotype frequencies are given as decimal numbers. Do not give genotype frequencies as percentages. It will cost you in the exam. From that, it then tells us what each of the individual number of each of the different phenotypes is inside the population. So of 500 organisms, we have 245, 210 heterozygous, and then 45 which are homozygous recessive. Again, if you don't know those terminologies, go back over the monohybrid and dihybrid inheritance presentations and pick up on those terminologies again, folks, because you do need them. What we're then trying to work out is how often in the population does the dominant Y appear, and how often in the population does the recessive Y appear. And that's what we mean by allele frequency. So if we look on the left-hand side equation, in the first red box, we've got 490 plus 210 equals 700. Well, the 490 comes from the 245 organisms that have 2 of the dominant alleles, so 245 times 2. The 210 come from the fact that the heterozygous, one of the alleles, is the dominant allele. So we have 210 alleles in that section of the population, which means that we have 700 alleles in that particular population. If we look on the recessive side, we look over to the homozygous recessive, and it says we have 45 organisms. Well, they each have 2 of the recessive allele, which is where your 90 comes from. And we've also got the recessive allele in the heterozygous, which brings in another 210, giving us a total of 300. Now, to turn those into allele frequencies, we simply work out the number of alleles divided by the total number of alleles available inside the gene pool population. As there are 500 individual organisms, and each organism has 2 alleles, there are 1,000 alleles in total. So we get 700 divided by 1,000 for the dominant, 300 divided by 1,000, which gives us 0.7 for P and 0.3 for Q. Now that we have a value for P and Q, we can work backwards. So if we have a value for P and Q, we know that P squared represents the homozygous dominant genotype. 2PQ represents the 2 heterozygous genotypes, and the Q squared represents the homozygous recessive genotype. And what you can see at the top of the page is a basic Punnett square with a monohybrid inheritance for a heterozygous set of parents. Again, folks, if you don't know that terminology, go back over the previous presentations on genetics. You've got to know these words. So we've got 2 heterozygous parents. We've got the allele frequency for each of the different alleles from the parents, P and Q, and we simply work out that P squared gives us a value of 0.49, which is the 7 squared, 0.7 squared. The 2PQ is 2 multiplied by 0.7 multiplied by 0.3, which gives us 0.42. And then we go to Q squared, which gives us 0.3 squared, which is 0.09, and that will give us each of the frequencies for the particular genotypes. What we can then do is go from there and say, well, if you've got these genotypes, these would be the representative phenotypes, and we would be able to determine if there were, say, if the question said there was 500 in the population, how many of those 500 would be homozygous dominant, how many would be heterozygous, or how many would be homozygous recessive, or we could even talk about it in terms of phenotype, if the dominant phenotype is seen or if the recessive phenotype is seen, by combining the homozygous dominant and the heterozygous values for the dominant phenotype and using the homozygous recessive for the recessive phenotype. We're going to have a look at some worked examples, but essentially that is what you'll be asked in pretty much every question of Hardy Weinberg. To start with allele frequencies and work through to a population in terms of genotype or phenotype, or to start with genotypes and phenotypes and work your way back to allele frequencies. It's always going in one of those 2 directions, and in some cases both. So when it comes to questions like these, there's a lot of information hidden in the question. You just got to be careful to make sure that you actually extract all that information. The LLT for tall pea plants is dominant. Well, you should know capital letters are dominant, but I'm going to highlight it anyway. It says there are T for short plants, lowercase. We've got a population of 200. 98 of those plants are short. And then we've got to work out the question. And the question is, what percentage of heterozygous. So I'm just going to put a Q there so I know that's what the question actually wants me to answer. If there's multiple parts, I'd put Q1, Q2, and Q3 just to make sure that I've answered all the parts that it's asking. So the first thing we've got is we've got 98 plants are short. Well, those 98 plants represent a lowercase homozygous recessive condition. In terms of the total population, what we can say is 98 divided by 200 represents, on the Hardy Weinberg equations, Q squared. If we do 98 divided by 200 in a calculator, it gives us 0.49 equaling Q squared. And then we get the square root of that, means that 0.7 is equal to Q. Well, that gets us part of the way there. We still need to work out what the allele frequency of P is. Without the allele frequency of P, we cannot work out the heterozygous, which is of course represented as 2PQ. The second Hardy Weinberg equation states that 1 is equal to P plus Q. Well, if I know Q is 0.7, then P must be 0.3. And if I know P is equal to 0.3 and Q is equal to 0.7, the heterozygous, again, if you go with your Punnett square, with your monohybrid, Q, P, 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 Q, P, Q, 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 you can see that we have 2 here, heterozygous, hence we get 2PQ, which means that we do 2 multiplied by 0.3 multiplied by 0.7. Now if I go back to my question, it actually asks for the answer as a percentage. So when I do that calculation, I get 0.42, then I multiply that by 100, and that gives me 42% of that particular population is heterozygous. So just to make it a little easier to follow, this is the exact same answer, just laid out slightly better. We've taken the homozygous recessive alleles, and that is a genotype called Q squared, because Q represents the allele. We know that 98 out of 200 organisms are Q squared. We work that back to get a value for Q. We know the equation, and we have to memorize the equation, P plus Q equals 1, which lets us work out P. And then when the question says what the percentage of the population is heterozygous, we get 2 times P times Q. Then we multiply that by 100 to turn it from a frequency into a percentage. And that gives us 42%. Let's have a look at another question. In this particular question, we're looking at fur color in mice, and we've got a brown and we've got a gray. Now it tells us that the brown is dominant for gray, and it tells us about population size, and it tells us about the number of organisms. What it doesn't do is it doesn't start off with any kind of indication of what the allele letters we're going to use are. So let's make it easy on ourselves. The dominant phenotype is brown, so let's start off by clearly defining that B is equal to the brown phenotype, and we're going to go with lowercase b is equal to the gray. We then start working our way through the question, and it says that we have 168 of those brown out of 200. It tells us that the brown is dominant and the gray is the recessive. Well, if we know that information, what we've got in terms of phenotypes is 168 brown mice, but that makes up the genotype of homozygous dominant and heterozygous, and there's no way we can separate those. However, if we get the 200 and we take away the 168, 200 minus the 168 tells us how many of the homozygous recessive organisms that we have. So we get the 168 from 200, gives us 32. We then have to say, well, if I've got 32 out of the total population of 200, and 32 out of a total population of 200 means that we have a value of q squared, and that q squared value is 0.16. We then take the square root to get q, which gives us a value of q of 0.4. If we know q, well, 1 minus the 0.4 will give us a value for p, which means that p equals 0.6. Now, just as a good habit, when you've got exam questions like this, you kind of scribble around a bit, I understand, when you're answering questions, but it's useful if you can highlight these kind of points so that whenever someone's going through your exam question, if you happen to make a mistake somewhere, an examiner can quickly and easily extract critical parts of the question and award you at least part of the marks. There's 3 parts to the answer to this. Now I've got P and Q, I can essentially answer any question they give us. It says, what is the predicted frequency of the homozygous dominance? Well, the homozygous dominance, of course, is our P squared. We know the value for P is 0.6 squared. So 0.6 squared gives us a value of 0.36, which we can give as a frequency, meaning it is 0.36. If we needed to turn it into a percentage, we could simply times by 100. Our heterozygous is 2pq, so again we go 2 multiplied by p, which is represented by 0.6, multiplied by q, which is 0.4. We pump that into a calculator, and that should give us a value of 0.48, which again, if we needed the percentage, we times by 100, but the question says frequency, so we don't. And the last one, the homozygous recessive, or q squared, which again, we get our value of q, which is 0.4, and we square it again, we've already worked it out previously, is 0.16. And just like the previous answer, I've just made it nice and straightforward so that you can work your way through and see all the calculations laid out in front of you. I've also given the answers in terms of frequencies and in terms of percentages. So practice question number 3, we're given 51% of a population carries at least one copy of the recessive allele. This one's a little trickier because when they say 51% of the population has the recessive allele, what they are talking about there is the 2pq, that's a recessive allele, and q squared. What that means is 49% of the population are represented by the group that's not listed, which is the p squared. So if we know that 49% is equal to p squared, we can work our way back. We divide by 100, gives us a frequency of 0.49 equals p squared. We get the square root of that number, and that gives us p. P plus q equals 1. So 1 minus the 0.7, which is p, equals 0.3, which equals the value for q. Now we have the values of p and q. The rest of the question, no matter what they ask us, is going to be straightforward. So we'll have a look back. The 51% of the population carries at least one copy. That's fine. What is the predicted frequency of the population expressing the dominant phenotype? Well, the dominant phenotype would be any organism that had p squared plus any organism that was 2pq, because p is dominant over q. We now already worked out the value for P and Q, so we can simply put those calculations together. And P squared should give us 0.49, simply that 0.7 squared. Again, good practice. Let's highlight P, let's highlight Q. Plus, we've got 2 times P times Q, which would give us a value of 0.42. And when we add them together, it gives us a frequency of the dominant phenotype as 0.91. Again, folks, just be careful. Don't be tempted to turn it into a percentage. The question says frequency, so we keep it as a frequency. Okay, 0.91 is our answer. And again, just like all the other examples we've been given, just let the answer out a bit easier for you to follow, a bit more straightforward for you to follow. Just be careful when you're answering your questions in the exam. The use of highlighters or even just the use of boxes in pen to show the examiner where you're putting in the critical bits of information in the calculation make the whole world of difference whenever people are marking your exams. Highlighting those bits, if you've got the final answer correct, brilliant, full marks, no problem. But if you've made a calculator error somewhere, or if you made some sort of error along the way, highlighting those key bits of information can actually increase your chances of picking up a couple of marks. If an examiner has to hunt through an entire page of writing to try and find your answer, it's just going to be so much more difficult for you to make sure you get all the marks you deserve. So make it easy for the examiner. And to example number 4. Remember, all of these questions on Hardy Weinberg follow the same process of working through from phenotypes and genotypes to get P and Q, and then possibly back to phenotypes and genotypes again, or starting with P and Q and going straight to phenotypes and genotypes. So in this question it says 1 in 1,700, and we're looking at cystic fibrosis. Calculate the frequency of the recessive cystic fibrosis allele and the dominant allele, and then calculate the frequency of non-cystic fibrosis sufferers in the population. Well, straight away, we're going to have to do part 1 before we can answer part 2, but just so I don't make any silly mistakes, I'm going to make sure that I put a Q1 and a Q2 in that question, just to make sure at the end I go back and check I've actually answered both parts of the question. In an exam, it would be very easy to do the first part, forget to go on to the second, and you've automatically lost potentially half your marks. So calculating the frequency of the recessive cystic fibrosis allele, well, there it tells us that cystic fibrosis is recessive. Not in the easiest way they could have put it in an exam, but they have clearly stated that it's recessive. They haven't given us any particular letter to use for cystic fibrosis, so again we can make our own up. If you were feeling brave and wanted to go with C or F, be very careful in how you write those. Again, the uppercase and lowercase need to be very different. I'm going to make it nice and easy on myself, and I'm just going to simply say the letter A and A. That equals CF, and that one equals what I'm going to refer to as healthy. Now, in my shorthand I'm working out, I've put H and CF, which is absolutely fine, but I need to make it very clear to the examiner what CF and H is in any answer that I give. So calculate the frequency of the recessive allele. Well, if 1 out of 1,700 people have it, that 1 over 1,700 is the phenotype homozygous recessive, which in Hardy Weinberg is Q squared. Again, we need to work it back, and we end up with 1 divided by 1,700 and then the square root of that number, and we get a value of Q of 0.0243. We should know that 1 equals P plus Q, which means if we get 1 minus our answer for Q, and we should end up with a value of 0.9757. Once again, just to make it nice and easy on ourselves, we're just going to highlight that part, and we're going to highlight that part to show us where we've calculated P's and Q's, and we're going to say that equals P. Now that we've got the value for P and Q, that's part 1 of the question completed. Part 2 is calculate the frequency of non-cystic fibrosis sufferers in the population. What we would then have to do is we need to do the calculation for P squared and 2PQ. If we put those numbers, so we square root P, then we add in 2 times P times Q, which is 2 multiplied by the 0.9757 multiplied by the 0.0243. When we add all that up, we should find the value of 0.9988. If I wanted to do that in a calculator, I could just do that in a calculator. But again, I run the risk that if I haven't broken the question down into parts, and these things can be worth several marks, if I push the wrong number in the calculator, very quickly I can cost myself marks. So it's up to you how much of this you want to do in the calculator. I would definitely say you should show P and Q, and I would definitely say at a minimum you should write down things like 2PQ and P squared, so you can show the examiner you knew what the calculation was just in case something's gone wrong. And then obviously your final answer is that. Just double checking, again, it says in the question that we have to calculate frequency and frequency, so we don't have to do anything about turning things into percentages. And once again, I've set out the structure here where you can see all the working out all the way through for all the different values and calculations. This would be best practice in an exam, but obviously in exam conditions, you've got to balance showing every step that you do with making sure that you have enough time to complete the exam. One of the things that a lot of students have found recently in the new exam papers is that time is a definite factor. So I would definitely show the first part of the equation where Q squared equals. I would definitely show the values for P and Q. I would show my second calculation, P squared plus 2PQ. I may be brave enough to punch that next line into a calculator. So I may just take a risk and say, okay, well, that whole part there, I'm just going to pop straight into my calculator. And when I pop it straight into my calculator, I should get that number of 0.9988 out. Always write as many decimal places as you possibly can, and then look at the question, see if it wants anything in standard form or if it wants anything rounded off to a certain number of decimal places or significant figures. But it's best practice to write down as many numbers as you can see on your calculator and then round it off if you need to. Again, just to avoid mistakes, because rounding errors are very easy to do, and if you don't show any you're working out and you make a rounding error, you can lose 4, 5, 6 marks. If you show all the stages all the way through, you may only lose 1. In the exam, they might ask you to just do a straight up Hardy Weinberg calculation, but it's not impossible they could build this into some sort of practical where they'd ask you to look at observations in a table and work out phenotypes. They could then go on and start asking you to pick a statistical test and work out populations, genotypes and phenotypes and so forth. In this particular case, because we're comparing observed and expected, and it's categoric data, you should be using a chi-squared test. So if it starts talking about Hardy Weinberg and stats tests, have a good read through, just be sure, but normally it is a case of you're going to be doing a chi-squared test. And then what it'll ask you to do is interpret the results of the chi-squared, and then it will bring in things like, well, why did the Hardy Weinberg data not match with the population data? And then you'd bring in the assumptions of Hardy Weinberg and say, well, in Hardy Weinberg it assumes no migration, etc. And somewhere in the question it might hint at clues to one of those assumptions. If it doesn't, it'll just be a straight case of if you could tie any of the assumptions to the question, then you'd get the marks. So just to finish this off, we're going to cycle back to the assumptions that we mentioned at the start of the presentation. The Hardy-Weinberg equations only apply under a certain set of conditions. So there's 5 pictures on the screen. Pause the video now and see if you can work out what the 5 different assumptions are without looking back at your notes, and we'll have a look at the answers in a moment. So pause the video now. So the 5 pictures are the assumptions, and the assumptions are: number 1 is no mutations. We cannot have any mutations. We can't have any type of selection. We have to have random mating. We cannot have any kind of preference in terms of mating. Population must be isolated. You cannot have a population where you've got migration going on. And lastly, we have to have a very large population. These equations are only applicable in large data sets. When you start going to really low numbers of isolated populations, these equations no longer hold water. So make sure you, when you're going through equations in Hardy Weinberg, if it comes up in the exam, it's very common for them to ask you name 1, 2, or 3 assumptions. So make sure you've got those down in your head. And that ends the presentation on the Hardy Weinberg principles.